crops. So, Professor Kavas, I think the floor is yours.
Thank you, Katrin. So thank you for good introduction. Today, my name is Musa Kavas. So I am from Ondokuz, Mayıs University. So today I will speak about the development of tomato. So actually, in this presentation, I will try to summarize my work based on some literature data. Actually, the CRISPR technology has been applied extensively to develop plants with enhanced traits, focusing on the improved food and feed quality, increased plant yield and growth, and also development of some biotic stress tolerant plants. So actually, this figure or this graph is prepared based on the data provided by AAB-PlantEd, and so showing the status of the plants developed by the CRISPR-Cas9 system around the world. Okay, so when we look at the detailed way in this graph, we can see that the most important character is studied by the CRISPR system is the biotic stress resistance. Because as you all know, the plant diseases created by bacterial, viral, and fungi. So it is very important because the plant disease can cause to reduction of plant yield. So sometimes it can be very devastating factors. So it is very important factor for plant production. So because of that reason, researchers prefer to use these characters for CRISPR studies also. Additionally, these characters is controlled by, sometimes controlled as a single gene. So this is single gene characters can easily knock out, and you can see the result of these knocking out by using just tools. For example, there is a little studies related with the drought stress, because drought stress is a quantitative trait. So you have to knock out, or you have to overexpress lots of gene at the same time. So there are a few studies related with the drought stress resistance by using CRISPR system, but there are many studies with the single gene characters. So these are the single gene characters, and these are the most important factor for plant production and plant yield. So there are, we can see there are lots of studies published in literature. In addition to the biotic stress, there are many studies, but these are not commercially available. But there are many research published in the literature. So these are related with the drought stress. As you all know, that drought stress is very important, especially becoming more and more important because of the global warming and the climate change. And so there are many studies completed in plants by using CRISPR system. In addition to drought stress, also there are some heavy metal response and cadmium stress related studies performed in CRISPR in plants. So CRISPR technology has been applied to wide variety of crops. Some of them, actually, most of them is stable crops such as rice, tomato, soybean, maize, and wheat. As you see here, tomato is one of the most studied plant in CRISPR system because it is a model plant for plant regeneration and transformation, and it is very important crops for the human consumption. So there are lots of studies performed in tomato. In addition to the stable crops, there are also studies in some minor crops, such as the strawberries and maybe cucumbers. So there are a few studies in these plants also performed by CRISPR system. So as you see here, tomato is very important crop. Actually, there is important in the human consumption. So and also this is a model crop for the transformation and regeneration studies. So it is widely used for the CRISPR editing system also. Okay, when we check the total production of tomato, actually the total production of tomato according to the Food and Agriculture Organization, so reached to 155 million tons around the world, and Turkey, India, and China are the biggest tomato producers around the world. So when we check the commercially available CRISPR-edited plants, actually there are a few plants. Normally, as you see in previous slides, there are lots of CRISPR-edited plants, but a few of them is available as commercially available. So, for example, there is non-browning avocado and amylopectin-enriched vaxicorn, non-browning lettuce, and powdery mildew-resistant grapes, and the less Persian mustard grains. These are some example of the commercially available plants created by the CRISPR system. In addition to these, also there is two important tomato commercially available tomato. These are developed by USA and the Japan companies. Both of them has the increased antioxidant capacity. As you see in my previous slides, tomato is very important actually crops because of easy to transform and regeneration. There are many studies completed by using CRISPR-Cas9 system, but most of them is related with the biotic stress because, as I said before, the plant disease can cause to huge loss of the tomato production. So because of that reason, researchers focus on the development of tomato plants to resist the biotic stress. So in this context, we can see that most important biotic stress agent is virus and fungi. So as you know that virus can use the host cell to multiply their genome. So they use the plant host cell machinery system to multiply their genome. So in classical breeding, researchers use deployment of resistance gene, but some pathogens, some virus easily discard this resistance gene system. So there is an alternative provided by CRISPR system, so we can knock out the susceptibility gene or S genes. So here there is some example. I will provide the examples of these studies. But if we check the literature, so we can also see that there are few examples for the development of the leaf structure. Also, there are good examples for the nitrification of tomatoes. Also, there are a few examples of the drought and salinity stress tolerance. So I tried to summarize some works related with the development of CRISPR-edited plants in the resistance for the plant disease. So most of them is related to the virus and the fungi. So this is the study I want to show that, for example, this is in the study Paramanik and his colleagues try to knock out two genes at the same time. One of them is Salmonum lecopersicum PLO, and the other one is Salmonum lecopersicum LO1 gene. So by knocking out these two genes, because these two genes is susceptibility genes, so sensitivity genes. So if they successfully knock out these two genes, and they create some resistant tomato plants, and they did some bioassay, and they good results after knocking out these genes. So they create some virus-resistant tomato plants by knocking out these two genes. Another example for this study is the knocking out the DMR6 gene. So this is published in 2022, and in this study also they tried to get fungi-resistant tomato plants. So they targeted the DMR6 gene. Actually, this gene is first reported in Arabidopsis thaliana, but in tomato there is an ortholog of this gene. This gene is also a sensitivity gene, so susceptibility gene also. So people in this study try to knock out this gene, and after that they obtain good result for the resistance. For the resistance of Pseudomonas syringe, Phytophthora capsicri, and Pseudomonas neuropersicans. So they obtain good result for the knocking out these two genes. So as shown in this slide, also there is another important biotic stress factor other than bacterial, viral, and fungi. So these are nematode, parasitic plants, and also insects. They can cause to yield losses in tomato and also other plant species. So because of that reason, we decided to develop some root knot nematode resistant tomato plants. Actually, these type of nematodes affect different kinds of plants, such as pepper, carrot, watermelon, and cucumber. As see in these figures, after infection of the nematodes, there can be some crown gall structure, and also there are some some nodules can be produced by these nematodes. Because of these nodules, the roots of the affected plants cannot effectively use water from the soil and cannot get enough nutrients from the soil, and because of that reason, the plants can affect negatively by the infection of root knot nematodes. So, according to the American Society of Phytopathology, this type of agents can cause to globally $125 billion because of this infection. So it is very important for the horticultural production around the world and in Turkey also. So to manage this type of problem, researcher and farmer use different strategies, such as they can prefer to use resistant cultivar. And because in resistant cultivar there is a Me gene, but as I mentioned before, this type of resistance can easily discarded by the pathogens. Also, Me gene is sometimes can break by using the root knot nematodes. So also there are different approaches for managing this type of problem. So some farmers try to use nematocyds. Because of that reason, for example, this one is a palmitic acid and commercially available palmitic acid. So some farmers prefer to use palmitic acid for the management of the root knot nematodes. And by using this information, we decided to develop some tomato plants and having increased capacity of palmitic acid. This graph shows the pathway of fatty acid biosynthesis in tomato. As shown here, there is a common enzyme responsible from the synthesis of different kinds of fatty acids. These are hexadecanoic acid and octadecanoic acid, and both of them is called as the palmitic acid. So we are trying to, in this study, increase the own content of the palmitic acid in tomato by using different strategy. Actually, in this study, we try to select best gene for the increasing amount of the palmitic acid. So by doing some in silico analysis, we found 4 different genes responsible of synthesis of acyl ACP2 So we call these, all of them is FAT A, FAT B1, FAT B2, and FAT B3. And by using CRISPR system, but here we use different CRISPR approach. Actually, Tom also mentioned about different kinds of CRISPR system available in the research. For example, to knockout, we used CRISPR-Cas9 system, but in this study, we try to increase and we try to overexpress these genes at the same time. So in order to overexpress these genes, we use CRISPR-Cas9 system. Also, we use CRISPR activation system, and we try to increase the secreted amount of the palmitic acid by the root of the tomato to combat root knot nematode. And as a result of this study, we successfully overexpress 4 genes responsible for the palmitic acid synthesis, and we use specifically used root specific promoter. This is the first time we tried. Actually, when you look at the literature, you can see that there are some promoters. These are constitutive promoters, so using for the CRISPR activation system. But for the first time here, we tried to use roots specific promoter for the activation of internal genes. And as you see here, we increase the amount of total palmitic acid content, sometimes 5 times higher than the wild type plants. So we successfully increase the palmitic acid content by using the CRISPR activation system. Actually, you can see the different result here, because these are belong to different construct. As I said before, there are 4 genes we targeted. So, for example, here we targeted 4 genes at the same time, but here there is just only 1 gene targeted in one time. So we successfully increased the amount of palmitic acid by using CRISPR-Cas9 system. So then we performed the bioassay. So because we have to be sure our systems work or not. So we performed some nematode inoculation by using these CRISPR-edited plants. And then after performing this assay, we measured the roots. And as you see here, there are some examples of the nematode infection. So for example, here, this is our CRISPR-edited plants, and this is the wild type bobcat plants. So bobcat is a cultivar that used in this study, because normally we are using the Crocker genotype for the tomato transformation, but the Crocker has me gene, so somehow resistant to nematodes. So we used bobcat. Bobcat has no me gene, and so has no resistance to the root knot nematodes. So as a result of the study, we successfully inhibit the growth of the, as you see here, this is the wild type. So this is the CRISPR-edited plants. We successfully inhibit the growth of the root knot nematode in the root of the tomato. So I'll skip this. So in another study, we target ethylene responsive gene. Actually, when we check the literature, ethylene responsive gene is a huge transcription factor family. Also, there are lots of members in tomato, and we select some of them based on the transcriptome sequencing data. So one of them is called as ERF42, and we successfully generate these type of plants. Actually, some of the ERF, ethylene responsive gene, responsible from the ripening. So when we knock out the ERF gene, so the ripening time change after the knocking out of this gene. So another result, actually surprisingly, we observed that these type of plants, so this is knocking out of ERF44, 54. So these type of plants showed increased resistant to root knot nematode also. Then we decided to use another ERF gene. So this is the ERF72, but unfortunately, when we knock out this gene, we observed that our CRISPR-edited plants decreased resistant to root knot nematode. So we can say that there is lots of different ethylene responsive gene. Some of them responsible from the somehow resistance to root knot nematode, but some of them is not. We have to check which one is responsible from the root knot nematode. So we can say that ERF54 is responsible from the root knot nematode infection. Another study is carried out by my lab is the development of the tomato resistant to tomato leaf miner. Actually, Tuto absulata is very important pest in my country and especially in the Mediterranean country. Actually, it may be more important because of the global warming. Because based on the global warming, the population of these type of insects will be increased. So some year these type of pests decrease the tomato production in Turkey. Sometimes 20% reduction in the tomato production. So it is a very important pest in my country. So there are different strategies used by researchers to develop these type of pests. So when researchers check the wild tomato genotypes, so this type of the tomato has trichome. Yeah, there are lots of trichomes both in leaf of the tomato. So, but these tomatoes wild type tomatoes. So by using this strategy, we are trying to develop some tomato plants resistant to Tuto absulata. So, but here we will use different approach. We called this CRISPR combo system. So in this system, at the same time, we will knock out 2 genes and we will overexpress 2 genes at the same time by using CRISPR-Cas9 system and the CRISPR activation system. Actually, this is an ongoing project. We get some good results, but we didn't evaluate the results yet. Okay, as I mentioned before, CRISPR system used in tomato in different research and different strategies. One of these is the people used to develop some tomato plants showing increased growth habit, and some people developed some tomato plants showing increased regeneration after transformation. So this is the good example for this approach. So in this study, people and researchers try to knock out some gene to evaluate the fruit shape of the tomato. So actually, they analyzed the tomato genome and they found different BZR gene. So there is at least 7 genes, but some of them is responsible from the development of the tomato fruit. So when they knock out this gene, so they observed that the fruit of the tomato is increased by knocking out these genes. And there are also different CRISPR studies in tomato. So, but some of them is related with the male sterility of tomato. Actually, male sterility is very important characters in tomato because most of the tomato cultivar used in the production called as F1. So F1 means these are hybrid plants. So by using the hybrid system, so farmer produce more tomato. So because of that reason, male sterility is very important because during the hybridization system or hybridization process, researcher must get rid of the male organs. So to create male sterility plants is very important for tomato production. This is the example. So this study published in 2020. So they knock out a kind of transcription factors, and then they easily produce some male sterile plants. Also, we successfully knock out some male sterility gene. So, for example, one of them is called as Solanum leucopersicum PhD type MS21. So we published this study in 2022, and we successfully knock out this gene, and then we produce actually 10 different lines. So some of them, you can see some of the results. So after knocking out this gene, we produced some unviable pollens. And because of these unviable pollens, as you see here, we observed our fruits our fruits has no seeds because there is no viable pollen. Because of no viable pollen, so we couldn't produce any seeds in our fruits. And another study related with the male sterility, we also knock out the aconitase gene. Aconitase gene is located on the Krebs cycle. So it is this gene has very important function in Krebs cycle, but also responsible from the development of tapetum, pollen tapetum. So we try to understand this gene is responsible for the male sterility or not. So we successfully knock out this gene. And as you see here, after knocking out of this gene, most of the plant, but not all of them, but most of the pollen has not viable. So, but if you look at here, wild type plant has lots of viable pollen, but our knockout gene has no viable pollen. So then we observed that some of the line has no seeds, but when we compare the male sterility 1 gene here, some of the line has seeds, so different from the MS1 gene. So another traits studying with CRISPR system is the fruit ripening. For example, here people tried to knock out fruit ripening gene. So there are many studies. So after knocking out of this gene, so people observed that the mutant plant reached maturity later than the wild type plant. So another application of CRISPR system in tomato is the development of drought and cold, heat, and salinity stress. So in this context, also we tried to develop some tomato plants for drought resistance. Here we targeted 2 genes. These genes are called as Solanum leucopersicum HP2 and HP3. These are responsible from the cytokinin signaling pathway. Actually, as you all know, that cytokinin signaling pathway and cytokinin signaling gene is very important gene because they are responsible for the plant morphology, plant development. So they has huge important function. But we tried because of based on the transcriptome sequencing data, we found some gene, maybe it can be responsible from the drought stress response. And we select these 2 genes and we successfully knock out these 2 genes. And we observed that our mutant plant has fewer stomata, and they showed lower performance when compared to drought stress. And if you look at here, there are also our genome-edited plants produce more lateral root when we compare to wild type plants, especially in drought stress conditions. But in control conditions, the wild type plants show better performance. But in drought stress conditions, our mutated plant showed better performance than wild-type plants. And also, surprisingly, we observed that the knockout plant. So we checked the abaxial side of the leaf belong to the mutant plants. We observed that our mutant plant has fewer stomata, and the stomatal aperture of the mutant plants is smaller than the wild-type plants. So because of that reason, I think because of that reason, they showed somehow increased tolerance to drought stress. So in another study also, we are trying to increase the drought tolerance of tomato by using CRISPR combo technology. In here also, at the same time, we will knock out a gene, and we will overexpress a gene at the same time. So we select two genes. One of them is called as AA18, and this is called as LBD16. So this is an ongoing project. So when we finish, I will report this also. And also, we successfully knock out some MET kinase gene. So because of time limitation, I don't want to go into detail. And also, we are doing some knocking out studies in long non-coding RNA. As you all know, non-coding RNA, long non-coding RNA is very important, especially for the controlling of gene expression. So to develop some drought resistant tomato plants, we are trying to knock out long non-coding RNAs also. This is the example, but we couldn't evaluate the response of these plants under drought stress. We maybe next year we will finish these studies also. At the end, I would like to thank you for listening my presentation. Also, I would like to thank to funding agency of TÜBİTAK and to my university. Also, I want to thank my team. Actually, in Samsun, in Ondokuz Mayıs University, my colleague and I studying with the genome editing. So my colleague, Associate Professor Kubilay Yıldırım, also studying on genome editing of plants, but he has project on genome editing of sugar beet and sunflower. As a last slide, so as Gerent also mentioned that we are organizing a conference in Samsun in Ondokuz Mayıs University. This actually this event is a continuation of the cost action of PlantEd. So at the end of October, so 29 October, so we will organize this meeting. So I cordially invite all of you for this meeting also.